Hello students, today we are going to discuss about the next term of your syllabus, that is the genome. Into the last lecture we have to discuss about the what is the gene. So gene, it is a structural and functional unit of the genome. So what is the genome? The genome, it is a genetic material of an organism. The genetic material of organism, the genetic material, it is one of the important factor or important specific part of bacterial cell or any living cell. They will be responsible for the hereditary and variations. This genetic material is most of DNA in case of the prokaryote as well as in a eukaryotic cell. So the specific DNA, it is double stranded, always double stranded in case of the prokaryotic and eukaryotic cell. So the genome consists the DNA, but in case of the some viruses, the RNA, it is also act as a genome. Some viruses like retroviridae like HIV, then also the specific types of the COVID-19, they can consist the RNA, it is also genome. So number of influenza viruses, they can carry the RNA as a genome. So the specific either DNA or RNA, but in case of the prokaryotic and eukaryotic cell, the genome is always double stranded DNA, but in case of the some viruses, RNA, it is also act as a genome. So the specific genome consists the double stranded DNA or RNA in case of the viruses only. Then it consists of both genes, coding as well as non-coding genes. So coding genes, they will be code the specific types of the proteins or proteins, and non-coding genes, they can't synthesize or the can't transcripted into the RNA, either can't translate it into the specific proteins. But the coding genes or coding DNA, they can synthesize the specific types of the proteins. So because of this, the coding genes are called as a cistrons. Why these are called as a cistrons? Because the specific types of the coding DNA, they have ability to replicate, transcript and translate. So because of this, the specific types of the coding DNA are called as a exons, or they are called as a cistrons. And these cistron, they have ability to cis and trans. Cis means they will be replicate, and trans means they have ability to transcript and translate. So because of this, the coding DNA are called as a cistrons. But non-coding genes are can't synthesize RNA, or also they will be unable to translate into the proteins. So these are called as a non-coding genes. So the specific genome that consists the coding as well as non-coding gene. Except of both of these, there is also one of the type of genome, that is a mitochondrial DNA. Mitochondrial DNA, these are ancestral bacterial cells. These mitochondria, they have their own genomes, and these own genomes, they can carry it also into the present era also. These mitochondrial DNA, these are also called as a mitochondrial genome. And also like a mitochondria, there it is a genome present in case of the chloroplast, and this chloroplast DNA also called as a genome, that is present in case of the plant cells, and mitochondria present in case of the plant as well as in animal cells. These mitochondria, they can carry the genome. This is also called as a mitochondrial genome. The specific chloroplast, they can carry the DNA. It is also called as a genome. Then the study of genome is called as a genomics. The study of genome, comes under the term genomics. The number of archaea and most bacteria have a single circular chromosomes. The specific archaea and most of the bacteria, they can carry the single circular chromosome, but some bacteria consist the linear and or multiple chromosomes, like eukaryotic cell, like a eukaryotes. The number of eukaryotic cell, they can carry the linear and multiple chromosomes, but bacterial cell, they can carry the single circular chromosomes. But the some bacteria also carry the linear and multiple chromosomes. So everything I have the exceptions, and like the same, the specific types of the bacteria, they can carry the single circular chromosomes, but some bacteria, they can carry the linear and multiple chromosomes, like a eukaryotic cells. So the specific term genome, it is carried by the scientist Hans Winkler in 1920. Initially, the term gene is described by the scientist Johnson in 1908, and the specific term genome is created by Hans Winkler in 1920. So gene, it is a structural and functional unit of the genome, and genome, it is a group of genes. And this term is carried out by the created by the Hans Winkler. So another important thing regarding to the genome, that is the genome size. So genome size, it is also important thing regarding to in case of the genome, the total number of DNA base pair in one copy of haploid chromosome. Here, the total number of DNA base pairs in one copy of haploid genome is called as a genome size. The total number of DNA base pair in one copy of haploid genome is called as a genome size, and it's variable in widely across the species. It is variable in different species of bacteria. It is can't same. Each and every living thing, they have the specific type of genes and the variable number of genes, and also the variable number of nucleotides present in their genome, present into their genomes. So the specific, that is a genome size. So for example, in case of the human, there it is a 3.2 billion nucleotides are present in a single genome. There it is a 3.2 billion nucleotides present in a single genome, that is near about size in 3 GB. That is a key 3 gigabytes. The near about the 3 GB or 3 specific 3.2 billion nucleotides, and approximately near about the 20,000 genes are present in case of the human being. That is Homo sapiens, that can carry the 3.2 billion nucleotides, or it is approximately 3 GB, and near about 20,000 genes are present in a single genome. Then in case of the E. coli, E. coli consists near about 4.6 into 10 raised to 5 nucleotides into their genome. That is a 4.6 into 10 raised to 5 nucleotides present in a genome, nucleotides present in a genome of E. coli. It is near approximately 4.6 MB. That is a 4.6 MB, and near about 88 genes are present in case of the single cell of E. coli. That is a 4288. There is a number of genes present into the E. coli cell, and near about 4.6 into 10 raised to 5 nucleotides. It is approximately 4.6. It is the size of E. coli genome, and 3.2 billion nucleotides, approximately 3 GB. It is the size of human genome. It is a specific genome size. And the last but least, it is commonly known as a group of genes. The specific size, or the specific group of genes are called as a genome. The group of genes are called as a genome. So genome, it is the most important term in case of the biology. That is, the genome size is variable in prokaryotic cell as well as in a viruses. The DNA or double-stranded DNA, it is a genome. It is a type of genome. The double-stranded DNA is a type of genome which is present in all bacterial and eukaryotic bacteria or prokaryotic and eukaryotic cell. But the RNA, it is also act as a genome in case of the viruses. And the most important, the term genome is coined by or created by Hans Winkler in 1920. It is about the second one concept of your syllabus, that is the genome. And then into the next lecture we will be discuss about the genotype. So thank you.